Hello, everybody.
Je sais pas s'ils m'entendent là, les gens. Si, ils m'entendent quand je parle.
We are going to start our roundtable on sharing and exploiting HTS data. Don't hesitate if you want to sit closer so that I will feel less alone. Okay, we had a lot of information about HTS during the week. We had a satellite event, we had a session. In nearly every session we hear about HTS, so let's now go a little more in detail of how to do and how to share and how to exploit. So I'm Valérie GRIMAULT. I'm the Assistant Director of EPPO, as a European and Mediterranean Plant Protection Organization. And we co-chair this session with Charlotte Trottin, who is also from EPPO and who is specialized on diagnostic and who has written with a panel of experts some guidelines on the use of HTS. And Charlotte prepared this roundtable with the members of the expert working group, who is currently working on our first HTS standard, HTS for virology. So thank you for them for helping for preparing this roundtable. So we are going to explore the limitations, the risks, the opportunities, how to share HTS data and how to exploit them. And we decided to do it interactive so that you work. So I will ask you to take your phone. We use a lot our phones this week. Take your phone, take the QR code, and like that, you will see the questions, which will be given by our colleagues, and you will be able to answer. So you will have questions where you have several answers, open answers, or just yes, no. Just before, just tell me if you don't have the QR code yet and you do not have the app yet. I still see people with their phones, so. Oh, the screen is moving. You can come here and do it here. No? Okay. Okay for everybody? We can start.
So good afternoon, everybody. So I'm Charlotte Trontin, and I will. So we have split the session in two. So we propose that we first discuss about sharing HTS data, and then second session on how you exploit HTS data. So and I just want to thank Samuel that is here to help us with all the questions that we will ask. So Samuel, you can switch to the first question. And this is just for you to get used with the app. So you still have the QR codes if you have not used it yet, and you can save from where you're from. And it will give us an idea of how many people are here. And so the last one, come on and scan the QR code and tell us from where country you're from. Just a way to get used with the app. So a lot of people from Australia, France, the Netherlands. France, I'm not surprised because we are new. Australia, I'm more surprised. Just that they are using HTS a lot. Okay, so it's nice. So we have a number of countries represented. And so I will ask Samuel to go to the second question. So we are going to talk about data sharing. So so we put a number of questions. It's just a way to get an idea of who is in the room and how you share data, and then we can discuss. So the first question is, do you share HTS data or part of them to the scientific community? No, there is no word. So it's quite, it's not surprising, but most of you share data, and some of them, not all of them, but there are still few people that share all of them, and some of you don't share data.
I just forgot to tell you that the roundtable is recorded, so when you will speak, you will need to take the microphone, and I will look if you need it.
Okay, so maybe Samuel, if we can go to the next question, which is when do you share your HTS data? So within a project, so within the researcher within a project or in a short framework, after publication or before publication? And if you, if there are several answers, you can select several answers. So we still get answer, but we are not surprised so much by the results. So most of you share data after publication and then within project, but we still have some people that share data before publication, and we have heard during the session on virology, during the satellite event, how important it can be to share this data before publication. So I hope that we can discuss it a bit further during this roundtable. Next question is, with whom do you share your data? So scientists you are collaborating with, scientists that are working in the same area, or the whole community? So the answer, so most of you are sharing data with the scientists you are collaborating with. Second answer is the whole scientific community, and the last one, scientists that are working in the same area. So we can see that you're more prone to share with the scientists that are in your close network than with the whole scientific community. So maybe we could first have some discussion with these 3 questions that we had, maybe to discuss what is limiting for you when you share data. Why do you prefer to share data with the people that you are collaborating with? Maybe it's a good idea to start discussing on that. So maybe if someone would like to take the floor to express. Yeah, I can see her. No? No.
Yeah, for me it depends. Like these questions is before and after publication. Like I would share it with the whole community, but after publication. But before that, only my own community.
Is everyone doing the same, or?
You mean with your own community?
Like my own research group or your group collaborators?
Microphone? Sorry.
Yeah, so if it was during a project, I would share it with my own research group or people that I work with closely. But then after publication, it's fine for everyone.
Adrian?
Yeah.
I think probably most of us are of the same mind, that the risks certainly of, on the one hand, being scooped, on the other hand, risk from a plant health perspective, risking trade impact of pre-publication sharing openly is perceived to be too high to share it outside of your closed network or your collaboration network before publication. After publication, it's public anyway, so it doesn't really matter. But I suppose I would then have a further question of, do we think that those perceived risks are actually real, or is it just us having that kind of innate mistrust of other people that leads to that? Because for me, I've got no way to gauge, because I've never released openly things, so I just don't trust people that they wouldn't scoop us or take action against us.
It's the end of the room.
So I guess it depends a little bit on the data for us, but our philosophy now is that we will upload data for the pathogen that we're interested, including raw reads and things like that, to a public database once we're published, but we don't publish the rest of the metagenomic database. Data, we keep that in-house, just because of risk of other people data mining and finding other things that we haven't checked the data for. And, you know, if it's proprietary sort of varieties and things that belong to, you know, groups like seed companies that might have, you know, genes of interest, things like that, we prefer not to risk that sort of risking confidentiality as well.
Anyone else? So maybe we can go on with the questions. So Samuel, next question, please. In your opinion, what type of sequence data should be shared? So if you share data, what type of sequence do you share? Raw reads, cleaned and demultiplex reads, annotated sequence, or you think that the sequence data should not be shared? So it's nice to see that it's between annotated sequence and raw reads, and actually maybe it also depends on when you share the data and the type of data you share.
Would you like to comment on it?
Thank you. For me, I'd like to see like the raw reads or the cleaned and demultiplexed reads, so that you can like either there was a talk on Sunday about somebody going back and reusing HTS data to look for other things, and also that way you can just double check and make sure that different pipelines line up and that kind of thing. So I think it just has more utility on the basic research end if it's like raw reads, rather than already been like gone through the pipeline and been worked on. That's just what I think.
Adrian, comments?
It depends. So again, I think there are times that I would happily share the raw reads, but in terms of open sharing, it would be the annotated sequences. With restricted network, I would be happy sharing raw reads. Once we publish, quite happy to put things on the short read archive. So probably all of those things, but at different times, if that makes sense.
So do you, once you have published, do you always put the raw reads? Or it also depends?
It depends, but pretty much.
Yeah, you underline here, because we are, I think, in a 4-dimension matrix, difficult to represent, but something like that, because it will be variable depending on what you put here. You could either share all the raw data or the annotated sequence, like Fiona mentioned, first dimension. Second dimension is with who. Someday this will be variable. Third dimension, at when, in fact. And the fourth, depending on the ownership. So it's like, well, we cannot represent mentally the 4-dimension, but it's like that. And so I agree completely with Adrian saying it depends. And it depends on when we put the cursor for these data sets on this multidimensional way.
Anyone want to comment on that?
You agree with it? You do the same? You have the same philosophy? I see heads nodding a lot. Okay, thanks.
So, of course, it's not only sequence that we share, but other data can be shared. So, to your opinion, what are the other data that should be shared with the sequence? So, none. Metadata related to a data set, a sample. Information on the HTS protocol used to generate the data, and others. So we have one other. So the HTS protocol seems to be a bit more important for you, but yeah, it's very close to the metadata. But I'm quite interested to hear about the one that click others. Maybe they can explain what other data they think should be shared. Adrian? Yeah.
Closer.
So I suppose it depends, again, what you term metadata, but for me, there's the contextual data which goes beyond metadata. So it's not just where was it sampled, when was it sampled, but the why was it sampled? Why were you HTSing it in the first place? What else had it been tested for? So that's kind of above and beyond the metadata for me, but that also gives the context to what's being reported.
Just while the microphone is moving for the new ones who arrived, you can download the QR code, and like that you can participate to our discussion by answering questions for our roundtable.
Yeah, I feel like all the methods used to prepare the data should be added to GitHub or some other repository, like a complete script pipeline, so you can recreate exactly how you did everything, and people can then go through the pipeline and recreate it for themselves.
I have disappeared.
It's like when you go to the cinema and the guy sits in front of you in a big hat.
So, I know EPPO, you never use must, but I would say metadata must be with the data. Must. Because without metadata, it's useless completely.
But it's interesting what you said, because so we had a workshop on HTS at HEPO, and we had someone from the medical field that kind of made a talk to open a little bit the perspective to know how it was used, and he was saying that they only share sequence and they never share metadata. So, well, at least on the sample. So it's interesting as well to see that there are differences with the field.
I am of the opinion that things will get better as we move forward, because new discoveries will be less and less important.
So with your comments on the medical researcher, I think they have metadata, because I think they're talking about humans, and that's like a host. And for us, that would be like a plant, for example. So that is already very important information. And they probably also know where this person lives or have been. So I think there's a lot of similarities. Yeah.
no one wants to comment, maybe we can move to the next question, which is on which database do you share your HTS data? So it would be a word cloud. So just put a few words on the app just to see how people share. If it's only NCBI. All these are kind of the same, right? NCBI and OSRA. Pre-publication spreadsheet. Yeah, so basically there is currently one database where you share data. Another one, bioproject, maybe the person that put this can comment. Yeah, QBank. So who put bioproject? Yeah.
Yeah, normally I put my data in short read archive, but also have a larger bio project for different components of the project, and also have a bio sample for each sample. So for me, it's NCBI, but there's SRA and bio sample and bio project within that. So it's like one submission within the NCBI.
Ah, okay. Who put the pre-publication spreadsheet? Yeah, Marlene, maybe you can comment on that.
So, some time ago, Sebastian and Adrian, myself, and some colleagues sat together, all with the same problem, that we all sitting on a lot of HTS data, like, yeah, everybody working with HTS has. It's getting a bit dusty on their servers, and not having the time or maybe the money to get it all published and properly characterized biologically and written up. So we've been thinking for a long time what would be a nice way to actually inform a trusted group of scientists, in this case virologists, and find out in what way we can share it in a pre-publication way. So we found a way to share the metadata and the finding information on what kind of new finding it is. This is a new finding for science, a new finding for a country or a host. And we actually tried this spreadsheet, filling this spreadsheet and sharing it among a group of people with, I think, I don't know how many researchers. And it works nice, but we're still updating that and refining it. And I think you can maybe add a bit more.
Yeah, so the idea was that if you have the metadata and the sequence data, you have sensitivity. So you either share the sequence data without metadata, or you share the metadata without the sequence data, and then either way you have to get in touch with the original posting scientist to actually get the full information, and then you can share peer-to-peer. And for us, that takes out some of that risk and nerves about somebody taking the information and running with it. So it takes out the trade risk because you don't actually have anything to compare your new clostridovirus and whatever with. But it also means that if we then want to go back and have a look at everything that people have put on the spreadsheet in tomato, or everything that comes from a certain region, or everything that came from symptomatic plants, we can go through and actually sift that out and then start contacting people to see whether we can work together on publications or to try and publish in just by putting material on GenBank or making annotated lists of this stuff and actually share it more widely.
And I'm not sure if it's pre-publication sharing. It's before publication or you...
Pre-publication.
Yeah, pre-publication.
But the idea is we get either publications or useful material out of it. So yeah, it's pre-publication, and that's why it's a limited group. It came out of the UFresco project. So it's the UFresco project plus some other scientists that were invited because they've been members of other projects we've been part of. And the idea there was that we could just use it as a test bed to see how it worked.
Yeah, question in the back.
Yeah, this is an excellent example of good collaboration. And as I mentioned before, as I feel that new sequences will not be as significant as they used to be or they are now, those type of projects will be able to provide us with high quality publications, and all of us actually will go into the spreadsheet rather than try to avoid it with all the information actually.
But for me, this result that we see now, it's quite interesting given some of the talk that we got earlier on diagnostic and HTS, because someone said that in NCBI you cannot really rely on what's there. So for me it brings a real question on everybody puts this data on NCBI and not on something else, and probably at the same time you have difficulties exploiting this data from NCBI because you don't know which one are reliable or not. So I don't know if someone wants to comment on that or develop further, but we can also probably develop it when we talk about exploitation of HTS data, because probably you have to circumvent this it in a way when you exploit data as well. No comments. So we will move to the next question, which is also a word cloud. So for you, what are the main limitations for sharing your HTS data?
This time it's a word.
Size. So it's quite interesting to have time first. I would not have expected this one. Confidentiality was expected, I guess. Biosecurity, we mentioned it earlier. Size. Misuse, theft. So maybe we can go with time first. So if someone wants to comment on why answer time, is it the volume or because the database are too time consuming? Yeah, the first item is time. So I was wondering what we mean with time, if for the one who put time. Is it a database problem or what is?
Or is it just time consuming because it takes time from your time to share that? I see her head's nodding.
I think everybody working with HTS now, also with the novel approach, will have the experience that you find a lot. As you may usually say, if you turn on the machine, you have like, in a week, you have like many new findings, new for your country, new for a host, new for science, but next week there's another week, you have new data, and then if you want to give the contextual data, the host, importance, the other viruses, other pathogens presence, the incidence, all the data, and getting it into NCBI, getting it published, or like prepare a manuscript, share the stuff with other researchers to see if they found the same thing maybe in their country. Like even for one accession, it takes quite some time. And if you have like all the time, it's like a waterfall coming in. Yeah. So right? Am I exaggerating?
No. No, it's like a waterfall, and you're standing with a small cup, trying to catch the water. But for me, as Maya said, I fully agree. And sorry, Marlene said, I fully agree. And this is, for me, the other side of the discussion we're having about biological relevance at the satellite meeting on Sunday, that we ultimately have to prioritise what we do these things with, because we only have a limited number of people and a limited amount of time. And so ultimately what you're doing is making, on your very early taxonomic IDs, you're making a gut feeling call on what may be the risks, and you're prioritising those for further work, whether that is publication, whether that is biological characterisation.
Say one more thing. Yeah, so for getting this information published in the way many like journals work, that in itself takes a long time. There's not an easy way to just upload a lot of like well-curated sequences with contextual information and some information why it is relevant and why we should care. There's not, at least not a very easy format I know of. And I don't know if anybody does. I would like to hear about it.
Our second item is confidentiality. So I guess that after publication, it's not really an issue anymore, or is it still an issue? Is it really limiting even after publication? I can see nodding at the end. Maybe you can comment.
Again, it's just around loading up entire sequence data sets and confidentiality of the information that might affect trade, might affect clients, all those sorts of things that are really important as well. So that's one of our big issues. And so, as I said earlier, we just try to load up the relevant information and nothing more. Yep.
But you did say that after publication, you are
There's certain
sharing.
data we still won't load up around metadata as well. I mean, we wouldn't want to identify a particular client if it's sequence that we're allowed to analyse, but they won't want to be identified. Those sorts of things are really, really important as well, because it can affect their reputation as well.
Yeah, of course. Yeah. Okay. So Marlene first, and then in the back.
So maybe a question for Fiona. With the we think that in many cases you just share and provide information you can share, you feel comfortable sharing. So in the case of confidentiality, we never will give the names of our clients or companies ever. So if you wouldn't do that, would there still be? Can you give an example maybe of a situation where you don't provide the name of company or the region?
It might be found in we might not give a locality, like we might say it's found in Victoria, but we wouldn't say it was found in a specific town. Those kinds of things that we wouldn't supply, for sure, yeah.
There was, yeah.
Just to go on what Fiona was saying, was that especially with data from when it's infected with a virus, so when you do the high throughput sequencing, it's got information all about the plants and plant genome as well. It's not like a just viral data, it's a mix of a whole heap of stuff. So in that regard, that would be considered, as Fiona said, if it was a specific breed of plant that has IP around it, then we can't load up the data that goes with the virus because it's also got plant genome in there as well.
Adrienne, you want to?
And picking up from what Fiona said, I suppose there's no real difference there between what we'd say putting a new disease report or a disease note, where if I said that we'd found something in a glass house in the southeast of England, then there's only one glass house that could be. So we kind of wind back and back until you get to a level that everyone knows who it is, but nobody knows who it is, if that makes sense. And interestingly, we did propose, several years ago, a short communication style report journal for sequence data, where you could put the annotated sequence and all of the contextual data. And we actually went round several journals and touting it as a potential, so an addition to disease notes, if you will. And none of the journals took us up on the idea. And part of me wonders whether we were at a point where there weren't enough of the community who appreciated the problem that we were seeing. And if we were to try something like that again, whether there'd be more traction. But certainly, I think at the time we saw what was coming down the tracks and thought that would be a good idea. And I don't know if there is a general feeling in the room if we should dust off that concept note again.
Adrian, I'm afraid whether something like that will be a dumpster fire of a situation, because people may just dump things without really curating them to the point that would have been publishable somewhere else. So the metadata is really difficult to curate, and if people think, not have, but think they have an easy avenue to put things out, you may not have enough capacity as a reviewer to go through the whole process. Just a thought.
Just for new people in the room, with the QR code, you can access the app which allows you to answer the questions.
So we didn't have other risks that were mentioned, but I think we kind of already covered biosecurity and misuse, trade issues. Maybe on size, I'm not sure who put size and if he wants to comment on that. No? Okay, so I propose that we move to the next question, and I will switch the next one because it was about the risk, and I think it's a bit similar with what we have already seen with the limitation. So we directly go with the next question. It's a question about Nagoya. Do you consider Nagoya when you share your data? Because I did not see us popping up in the previous question, and so. So most of you don't.
And I have a question because for me it's not clear. Is it clear for you? Are these data concerned by Nagoya or not? For me it's really not clear. Well, it's never clear for Nagoya for me, but
maybe the people that answered yes can comment. So if they consider Nagoya, probably they can explain. Yeah, Adrian?
So my understanding at the moment is that sequence resources aren't covered by the Nagoya protocol, but we consider the principles of the Nagoya protocol. So we would want to have permissions from, if something were sourced in another country to ours, we would want to have the permissions and preferably scientists on board from that country before we shared data or made things public.
Also for import inspections?
Import inspections are different. But still, if we're reporting something related to a country where it's never been reported before, we will still try and engage with scientists from that country before we publish.
But this is independent from Nagoya, or for you, it's independent from Nagoya? Or,
yeah, it's principles.
Principles, yeah. So anyone want to comment on Nagoya further? It's no... Okay, so we can move to the next question. So it was more for me, so we work for EPPO, so we are mostly interested about quarantine tests. So I was wondering if you always consider or look for quarantine tests when you share your data before sharing them.
You mean other than viruses?
most of the people do, but the no is also several people answered no. So I don't know if somebody want to comment. When you, yeah, Sebastian, go ahead.
Yeah, I put no because viruses, if I look for viruses, I will check for it. But we can also, in the transcriptomic data that we generate to detect viruses, have other pests that we do not systematically look for. We could have traces of other quarantine pests inside that we have overlooked.
Okay, so the one that answered no, is it a similar case as Sebastian mentioned, or it's just that you're not interested by quarantine, so you do not check in your research? I don't know. Anyone want to comment? And the one that yeah, in the back.
I was thinking, like, you check for what you're looking for, like a particular virus, but we really don't have time to then scour all the data for all the quarantine pests, especially not on the list from Australia. You just wouldn't have time, and if you're only looking for viruses, you do a blast with the data to the virus database, but I'm not going to be looking for fungal infections or bacteria or anything like that. You just don't have time in the project, and you just don't have the resources.
And just to add to Fiona's comment is that that's exactly why we don't release the data holus bolus. We only upload the things that we've looked for and we have permission to release.
I agree with everything has been said, but being a virologist and more on the molecular biology side of things, if my lab messed things up, or if the sequencing, place sequence things up, I would not be able to figure out what is happening if it's not a virus. If it's a virus, it's easy to see. If I start looking for bacteria and fungi and I don't really know where it comes from, it will take my whole time.
I think we will soon move to exploiting the data, but I had one last question that I wanted to, so there is no question, I just wanted to discuss how we can facilitate then, based on everything that has been said, how can we facilitate data sharing for you? And if anyone has some ideas to facilitate data sharing, and because right now for me, which is really surprising, is that everybody put his data on NCBI SRA, and at the same time, everyone criticized NCBI for not being reliable. So for me, it's very surprising.
I'm not sure I understand why you're surprised, because if you want to publish in a peer-reviewed journal, you need to submit your sequence data and some level of information there, so you have no choice. And I think there's a lot of, most of it is really, really good on NCBI. You just have to be wary of the stuff that's not good, and you need to be trained in looking for that. But I think it's a really great system.
So it's more a problem of exploiting data. So it's good. Take us to the second, right? It's when you exploit the data, being able to,
What does it mean, explain?
well, use the data. Being able to distinguish the ones that are reliable from the other ones.
Go to the publications and decide for yourself.
Are you asking as EPPO or as a member of the community? In other words, are you proposing that somebody at EPPO may help the whole process? Okay. So I have nothing for you.
No, I think it was more a general question about what would you need, or would you need something, or in fact it's easy to share data and you don't need anything except time for doing it.
So in medical field during COVID, but also before, GIS-8 became popular for some time, just because of the same reason, because people seen hurdles of confidentiality and also of being scooped if they share their data. So this reason can somehow be overcome with platform like this. So GIS-8 kind of asks all of the users to adhere to the policy of contacting and attributing the contributors if using their data. But it also had some problems, so there were also some affairs where people actually scoop the data and use it. So it was not ideal, but maybe just a thought if something like this could be useful also in plant pathology in general, or just plant virology.
So let's move to the second. Yeah, Valérie will share this second session.
So now it's for exploiting HTS data. So we can just move to our first question. So do you exploit HTS data which are available in databases, which means which are not generated in your lab? Data. Sequence data. Big majority of yes. So when it's no, does it mean that you have enough from your own lab, or you don't need from other for having data in databases coming from others, or why did you answer no? Just to know. Yeah.
I'm a bit confused with the questions. Sometimes with do you mean sequence, like annotated sequences generated using HTS, or do you mean the raw, the short read archives, or raw data that are uploaded to the system? So I use the former. I don't use the latter much.
Ah.
But maybe I think you do.
Well, we didn't make a difference between the type of data here. It was just more in general. So apparently it depends of the type of data. For some data you just use your own ones, and for others you go for information in databases. Yeah.
Yeah, I think most people were probably assuming SRA, but in my project I've gone through SRA data that a lot of them weren't used for virology, and just gone through, and you can sort of chew through the data again, which is why I was advocating for raw data sharing, because it is quite useful. But a lot of the time, if people haven't been clear with the metadata, it can kind of not be very useful. But I have definitely exploited it, the SRA data directly. But I would say that probably not many people here haven't used HTS data at all, if you're talking about annotations and stuff. Because I think most people here would have used them at least once.
Similar to what was said before, I said no only because I wasn't sure. Like, I'll use NCBI and sequences that have been done through high throughput sequencing that are put on there. But if you were talking about going through someone's raw reads or something like that, then no, I haven't.
Okay. Okay, we can move to the next question. So for what purpose do you exploit data? Is it for fundamental research, applied research, diagnostic, or product development? What? Wow. So mostly for diagnostic, but also quite a lot for fundamental and applied research, but very, very few for product development, maybe also due to the people who are in the room. So in fact, it's most for all the aspects of our jobs. Nice to see the diversity. I don't think do you want to comment, or is it the need? And do you sometimes exploit the same data for the different purposes at the same time? Or? Yeah, I see.
So if we're looking at, say, a novel virus, we will then search the SRA and try and do either a mini host range by electronic sequence searching, or at least a distribution, if we can. But quite often, the crops we're dealing with, there's so little data for those crops on the SRA anyway that it becomes a bit of a pointless exercise at the moment. But it will be interesting in 5 years' time to go back and do the same thing again.
Mm. Okay, we can move to our next question. So what are the main limitations for exploiting these data? You can answer with words. Oh, big time. Time. Is it linked to also the limitation for that we saw earlier? There are so many data that you really lack time to exploit them. Can you share some experience or give a little details of your experience?
For me, it's capacity, human capacity. When I started 15 years ago, there were probably 25% more virologists in the country. And then you have the same amount of work, if not more, with less people, therefore less time.
So we generate data, but we don't have the human capacity to analyze them? I see a lot of heads and a lot of sad faces. Yes, Sébastien?
I wrote intermediate database because, in fact, there's been a boom for RNA viruses from last year when there was a group of informaticians just mine all the SRA data to make an intermediate database with a signature of RNA viruses, which was super easy to mine by any scientist. And from that, there is really a boom in exploiting this data. And so, there is no technical, biological possibility, but if you have this kind of intermediate database which speed up and ease the work, even if we are 25% less, but if it becomes much easier to mine the database, to work with the data, we could handle it. We would welcome recruitment, but that's a bit this kind of element that I put there. This facilitated the mining of gigantic amount of data.
Okay, and how could and that would be one of the solution? And can it be applied to everything, or
maybe
what would be
not,
the need?
maybe yes, but it's at least for the RNA viruses, it has been really a boom.
Okay.
In 1 year. I mean, it is really a before and after. It was really a big milestone in the mining of database. Now everybody, any lab can mine all the RNA viruses that are in the huge database of SRA.
Does it mean that we would need that in other disciplines? I see a lot of virologists here also, from what I know you. Okay. Other experience that you want to comment about? Well, quality is quite popped up. Do you have difficulties with the quality of the data that you find? Yeah? At the end, yeah.
From my experience, quality tags with the discussion that we were having before in terms of it takes extra care to check that the data we're mining and we're exploiting we can rely on it. So from my experience is that mix of internal and external data that you start exploiting. So you start a little bit confirming each other as you process them, and then it's mostly making sure that the quality of the data you're using is good enough and you can rely on it. So that's why I put as a limitation quality, because you need to spend a little bit of extra effort, extra time to make sure that everything is done correctly and that you're not using wrong data or just data that points in the wrong direction.
Okay. No other comments, so I think we can move to the next question. So what are the risks of exploiting the data of others? You can also answer with a word. Okay, I'm not a specialist in HTS at all, but from what I see, you've put different words, but does it rely always the quality of this data that you don't really know? Or is it something else? You have spoke about uncertainty, reliability, cross-contamination, incorrect conclusions. Is it linked to the quality that we were talking just before? I see some heads. Okay, and the lack of metadata? Yeah, Adrian?
So I think many of these points are actually wrapped up together. That because you don't have the direct knowledge of the work that was done, the way the samples were taken, and how that relates to metadata around the sample, you then have the uncertainty about the reliability of the data. So yeah, that question, you've got wrong host or environmental RNA contamination. You know, I'm quite sure that quite a lot of the stuff that's being flushed out of some hosts is just, say, pollen on the leaves that isn't being flushed off. And that then means that is the host that it's, even if you have host data, can you trust that that pathogen was in that host, or was it on that host? And that takes us back to the biological relevance discussion. But because you don't have the sample in your hand, you've no way of being able to rectify the biological relevance question.
So shall we say that for exploiting data from others, we would need them to really give as much metadata as possible, which means if I have to share my data, I would also need to think of the other ones who are going to exploit them and put as much metadata as I can? Okay.
I don't know if that's really the solution, because I think data sets, also SRA data sets, are being exploited that were generated for completely different purposes than, for example, the purpose we would use it for, like testing maybe one plant with viral symptoms to see what virus is in there. Because we also exploit RNA-seq data for metagenomic studies while they were looking for something completely different. And they would have provided all the relevant metadata, but just not the one I'm interested in. So it's
difficult.
Yeah, it's difficult.
Adrian?
Well, and again, and that then leads on to the things that people like Fiona are worried about with the trade consequences of those findings.
Yeah.
And at one level, it can lead you into kind of blind alleys, or at the other level, the data you're looking at just seems like nonsense. You know, you find a plant virus in a human medical sample type thing.
I think all people in this room will agree that it's better not to publish than to retract. I have had the luck of trying to publish something, and I went in the database to find the sister virus, and they did not even put the genome in the right orientation. So if you have those type of things on NCBI on curated databases, what are you going to get out of that? And pollen will be the number one thing that it's really a mystery for me how people do not really think about pollen.
I think there will always be some uncertainty, no matter how much metadata we put on. So probably I think we really need is some kind of code of conduct. So if we are exploiting the data, which was not made by us, but we got it somewhere, how do we annotate it? How do we communicate it in the publication? How do we make it very clear that this is a valuable piece of a puzzle to get to know something, but that it can have its limitations? So maybe I don't know, a kind of some kind of guidelines to which things do we need to report when we are using this data could be very useful here.
Mm-hmm. Okay, well, we can move to the last question.
It's an open question.
Ah, it's an open question, désolé. So do you have ideas to facilitate the exploitation of HTS data? How could we help? Well, how could we as all of us, how can it be helped? What would be necessary to facilitate exploitation? Adrienne has a question, would like to answer. Oh, she's good.
I think Dennis has hit the nail on the head. For me, it's how we get some kind of code of conduct guidelines that we can then get people to at least acknowledge that there should be some level of responsibility from ourselves before we use that data. And yeah, I'm not sure how we do that. I'm probably too much of an idealist to be the kind of person to take it forward, because I think everybody's honest. But certainly something I'd be happy to pitch in and help with if there's a group of people who want to try and put something together.
Mm-hmm. So that's maybe somewhere where HIPPO can help. Other ideas to facilitate exploitation?
Hello. So for me, what we are missing in diagnostics, for sure, is a project like it was ValiTest, where we tested different methods and approaches, and then we did test comparison studies. And we should do the same, at least in virology, maybe also in some other disciplines, difficult to say for me. Again, what said Denis and Adrian, we would need some guidelines how to prepare data and how to use data. And well, actually this is also a political question, so we should incorporate also FAO, let's say, to give the instructions or to prepare the instructions for countries how they can prohibit it, let's say, import or export, what kind of evidence they need that there is really a new pathogen, not only on the basis of some sequences which somebody can find somewhere. If we will go in this direction, we will not share data at all in the end. So this is really, really important. And I believe that EPPO can do much in this direction, but of course other organizations, regional organizations. So this is actually first political question, and then of course we can also harmonize what we are doing. It does not mean that we need to do everything the same, but that we can find the protocols which we know they are working, and also the instructions how to use then the data, share it and use it.
Nice idea of possible future projects together. Harry?
Yeah, I have a concern about the beginning, and so we did a Tobamo-virus project with seeds. So with Tobamo-viruses, it's a challenge to get not cross-contamination, so I think you really need to think about appropriate controls during sampling and the processing of the samples in the lab. So in the front side, I think it's very, very important to have kind of a good laboratory practice, so that you will interpret the data in an appropriate way. And I think that will be quite a challenge. So there are big chances, but also some challenges about what to do with low numbers of reads.
Well, I think we have several members of the expert working group who is drafting the standards on HTS for virology, so you know what will be important for you to describe in your standard. But that's true. Other ideas?
Yeah, I think partly we all agree that it's important to share the data, but if we want to share the data, then we need to put somewhere where we all access, right? So if that's the case, then we need to put, for example, if this room is where we store our data and then everybody can come and share this data, then we need to put a guideline at the door. Anything that come into this room should follow that guideline in such a way that you don't just upload or you don't put in that anything that you just basically find because you find them. If you see NCBI, I'm not complaining, but there's a lot of really, I don't want to use the bad word, but it's garbage data there as well. There is quality data. But so how can we put something in place that NCBI, for example, put a guideline that anybody who upload data can follow that standard and then put the data there so that we can go and then have some sort of trust in the data that's in the NCBI, for example.
Mm-hmm.
I'm just a bit confused. All this garbage data and everything, isn't it the job of the journal or the reviewer to do the job, actually? To refuse the paper or not accept the paper? What I'm saying, like,
That's my point. I mean, if we all speak loud, so we don't have to make a bad round.
No, it's because it's recorded that we need a microphone. Okay. Okay, so if I try to wrap up what we have said today. First, really, a very big majority of you is sharing data and also exploiting data, not only your own one, but the ones of the others. From what I understood, sharing the data is really depending of what type of data, at what moment, before, after publication, for who, your close network or completely public, the type of data. From what I understood also, the availability of metadata is important. What you need is quality of the data. From what I understood, you need guidelines just to define how to reach this quality. We could have a nice research project like Validates that we could develop just to know how and to validate some protocols data, to validate some methods, and also to define guidelines. Did I forget something? Certainly.
Instructions of Maya.
Huh?
Yeah, I believe that it's a good approach, and actually what we did, Sebastian as the first author and many others prepared the guidelines how to deal with the data, the virus data you found in your databases, and this kind of protocol that was I talking about also needs to be on the political level. This is it was done. Researchers done the job, but now it needs to go further on. But yes, for project, I agree. In Valitest, we operated and methods were prepared by labs with quality assurance on place. So this will help with the problems like contamination and how you deal with data and so on. This is really very important because if you have proper controls, then you can evaluate your data quite well.
I don't know by heart the instructions for HTS, which are already in the standards at EPPO. But do we already have these things about controls and? Yeah.
No, I was just say about the code of conduct for exploiting data, and I think that's something that being done under the guise of either a regional plant protection organisation or even at the higher level by the IPPC would have a lot more weight to it, and especially when it comes to what's an acceptable amount of evidence to take trade action. And I think that could then feed back to calming a lot of the anxiety about sharing data in the first place.
Yeah. Oh, many hands at the back and after here.
So I've heard that there's a standard regarding sample preparation, alien controls, and such. I was wondering if there are any perspectives on data processing, bioinformatics, and interpretation. Is there a standard or a way of standardizing, harmonizing that part of the problem?
Charlotte? What is yet?
The only thing I can say, so for now we have not planned really to work on that. At least it's not on our work program. There are some guidance already in the work that has been done with Valérie tests and that we have used in our EPPO standard. But I know that in the last workshop that we had at EPPO, where we also discussed HTS, it's true that it has been a point that has been raised, that maybe more guidance about how to really perform the bioinformatic analysis was needed. So this is something maybe that we have to explore further.
Oh wait, there was Fiona first.
So just to bring it back to data sharing, there is a project in Australia at the moment where we're developing a genome reference database for traceability for outbreaks and things like that. And within that, one of the things they are doing is drafting a data sharing agreement between the various agencies that would contribute to that. And that genome reference database also sits alongside our reference collections for all our pathogens as well. So there is some work already sort of going on in that space that might be really useful. So I just thought I'd put that out there.
We had a question here, and after it will be Adrienne. Yeah, there was there first.
I would like to answer the question about the protocols for data and the whole protocol of the HTS. Oh, exactly. That's why we need a project, because it's a lot of different platforms. It's a lot of different possibilities how to do it, and we need to specify the ones which are working. And this is not what EPPO was done, because it is early stage of technology, especially in diagnostics. So that's why we need that. Of course, databases are really, really very important. So if we can have initiative more global, that would really add to the result in the end. But we need to work on different methods from the start, from the sampling, extraction, library preparations, and there will be for sure more than one. It will be many possibilities, but they need to be verified and tested in test performance studies, and then we have possibility to prepare guidance. So we are not there at the moment.
Excellent ideas, but I will go back to my favorite subject, which is regulations. And the problem are not necessarily that people don't understand, is what people's directives are. I've been in places where there's a clear case of a disease in front of me that is very easily identifiable, and the person on the other side says, no, it's not what you think. Off the record, said I was unable to publish it. So where am I going with that? Independent of all the good data that we can produce, we need to have a clear communication channel with regulators and really get a buy-in of what exactly they want in order for us to deliver. I cannot believe that there are countries still that are asking for bioassays where there's plenty of evidence that they don't really do much. Yet, this is a way for them to stop the material to get into their country because that gives benefit to their local producers. So this is an EPPO. This is whoever else is up there in the regulatory arena to really help us understand how to bypass what I call the crazies, the people that know what is happening, but they play it crazy so they will not really upset their stakeholders.
It was just to say that several of the things that have been mentioned here, so things like harmonization of pipelines, curation of databases, and how do we stop the crazies, are all things that came out on post-it notes during the satellite workshop on Sunday. So we can always look to pick up some of these discussion points under the Vernet 2 project with Ufresco, and then explore those discussions further and see how we can move this forward.
Yeah. Okay, that was a very nice conclusion. So thank you very much for your attendance and using the app so fast. I was amazed how fast you were answering the questions. Thank you for the nice discussions we had. Thank you for the microphone. I think you did around the room many times.